Welcome to the October issue of the AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. Unless you live on the equator, October is a month of marked variability. In the same way, our October issue is filled with a variety of great cardiovascular research and spans from the molecular to organ to human physiology. I know you'll enjoy what's inside, so let's take a look.
Angiogenic et al. Performed cardiac mapping with monophasic action potential Map Amplitude. They hypothesized that Map Amplitude, or MAPA, will provide more accurate infarct sizes than other mapping modalities via increased sensitivity to distinguish healthy myocardium from scar tissue. Using a rat MI model, they investigated the accuracy of in vivo ventricular epicardial maps derived from MAPA and other electrophysiological parameters versus infarct size. MAPA decreased between healthy and border tissue and border and scar tissue. Contrary to their hypothesis, BVA mapping performed most accurately regarding quantifying infarct size. MAPA mapping may have high spatial resolution for myocardial tissue characterization, but was quantitatively less accurate than other mapping methods as determining infarct size. MAPA may hold potential value for precisely distinguishing healthy myocardium, border zone, and scar tissue in diseases of disseminated fibrosis such as atrial fibrillation. Ayun and co-workers show that pressure overload-induced heart failure is exacerbated when nicotinamide phosphoribosol transferase, or NAMPT, is knocked down or overexpressed in a cardiospecific way. The NAD level declined in NAMPT knockout mice under pressure overload. In cultured cardiomyocytes, NAMPT knockdown diminished mitochondrial NAD content and ATP production, suggesting that downregulation of NAMPT induces mitochondrial dysfunction. On the other hand, the NAD level was increased in transgenic NAMPT mice at baseline, but not during pressure overload. The expression of SIRT1 was increased in transgenic NAMPT mice in association with reduced overall protein acetylation. Pressure overload-induced downregulation of metabolic genes was exacerbated in transgenic NAMPT mice. In cultured myocytes, NAMPT and SIRT1 cooperatively suppressed mitochondrial proteins and ATP production. Thus, endogenous NAMPT maintains cardiac-specific and metabolism function in failing hearts, whereas NAMPT overexpression is detrimental during pressure overload, possibly due to excessive activation of SIRT1, suppression of mitochondrial function, and upregulation of pro-inflammatory mechanisms. Johnson et al. Investigated the angiogenic activity of exosomes from induced vascular progenitor cells as a cell-free therapeutic approach for ischemia. Nanoparticle tracking analysis revealed that exosome isolates fell within the exosomal diameter, about 150 nanometers. These exosomes contained known markers, ALEX and TSG101, and their morphology was validated using transmission electron microscopy. When compared with rat aortic endothelial cells, IVPCs significantly increased the secretion of exosomes. Cardiac microvascular endothelial cells and aortic ring explants were pretreated with rat aortic endothelial cell exosomes, or IVCP exosomes, and basal media was used as a control. IVPC exosomes exerted an in vitro angiogenic effect on the proliferation, tube formation, and migration of endothelial cells, and stimulated microvessel sprouting in an ex vivo aortic ring assay. Additionally, IVPC exosomes increased blood perfusion in a hind limb ischemia model. Collectively, these findings indicate a novel cell-free approach for therapeutic angiogenesis. Tvita and colleagues hypothesized that hypothermia and rewarming induces left ventricular contractile dysfunction due to phosphorylation of cardiac troponin I at serine 23 and 24. To test this hypothesis, the response of wild-type mice to hypothermia and rewarming were compared with transgenic mice expressing slow skeletal troponin I that lacks the serine 23-24 phosphorylation sites. At baseline conditions, there was no significant differences in LV systolic function between wild-type and transgenic troponin I mice, whereas measurements of diastolic function, isovolumetric relaxation constant, and end-diastolic pressure-volume relationships were significantly reduced in transgenic skeletal muscle troponin I animals. Immediately after rewarming, significant differences between groups were found in cardiac output, stroke volume, and the preloaded recruited stroke work. However, end-diastolic pressure-volume relationship and tau returned to control levels within 1 hour in both groups. They conclude that hypothermia rewarming induced LV systolic dysfunction results from phosphorylation of cardiac troponin I at serine 23 and 24. Pepin et al. Examined whether the epigenome, specifically DNA methylation, is reprogrammed in heart failure to potentiate a pathological shift in cardiac gene expression. They used paired end whole genome bisulfite sequencing and next-generation RNA sequencing of left ventricle tissue obtained from 7 patients with end-stage heart failure and 3 non-failing donor hearts. Differential methylation was localized to promoter-associated cytosine phosphate guanosine islands, which are established regulatory regions of downstream genes. Hypermethylated promoters were associated with genes involved in oxidative metabolism, whereas promoter hypomethylation enriched glycolytic pathways. Overexpression of plasmid-derived DNA methyltransferase 3A in vitro was sufficient to lower the expression of numerous oxidative metabolic genes in H9C2 rat cardiomyoblasts. They identified binding site competition via hypermethylation of the nuclear respiratory factor NRF1 motif and established upstream regulator of mitochondrial biogenesis.
Thank you for watching this edition of From the Editor's Desk. As always, visit the journal online. We welcome your feedback concerning our review process or any other issues related to the journal, and don't forget to send us your best work now. Time to first decision is approximately 18 days. So keep that in mind, and we'll see you next month.